Molya disease of wheat. In this e-lecture, we are going to study about the symptoms, morphology and life history of causal organism, disease cycle and control measure of another nematode disease which is Molya disease of wheat. Let us start with the distribution. The Molya disease of wheat is also known as cereal cyst as it is caused by the cereal cyst nematode. Local people call this disease as Molya. Molya means deformed. In this disease, the root system is completely deformed. The disease was first discovered by Kuhn in 1874 in Germany. He first observed the sister nematodes on cereals as parasites. In India, the Molya disease was first discovered from Sikar district of Rajasthan in 1958 by Vasudeva. This discovery proved as a landmark event in the history of nematology in India. This disease is prevalent in light sandy soil of northern India region, particularly in the states of Rajasthan, Haryana, Delhi, Madhya Pradesh, Punjab and Uttar Pradesh. Wheat and barley are the two main hosts of the cereal cyst nematode. The affected areas in Rajasthan usually suffer up to 50% of crop damage. Main symptoms of the disease. In newly attacked field, the disease appears in small patches of about 2 to 3 feet in diameter. And if the same crop is cultivated year after year in the same infested fields, then these patches gradually increases in diameter every year until the whole field gets infested. The main symptoms of this disease are stunting of plant, discoloration of leaves, tillering is markedly reduced. Tillering means formation of side shoots. And in severely attacked plant fails to produce the ear heads. The infected plants are stunted, pale and looks unhealthy. These symptoms which appear on above ground part of plants resemble with the symptoms of nutrition deficiency. The main symptoms of the Molya disease appear on the below ground part of the plant, that is on the root. The roots of infested plants appear smaller than the healthy plant. Roots become typically bushy with slight swelling appear on the site of nematode infection. In badly affected plants, the whole root system becomes dwarfed and matted. As we see in this picture, the root system of affected plant become dwarfed and look like a mat. In the advanced stages of infection, that is by the end of November or in early December, the mild swellings appear near the root tips. This condition appears normally within 4 to 6 weeks after sowing of wheat. Glistening white bodies of females are also seen adhered to the root by the mid of February, and this is the diagnostic symptom of this nematode. These bodies of the female nematode become brown and they may remain either attached to the roots or they fall into the soil. These dead bodies of the females are known as cysts. These cysts are filled with the eggs and they are the main source of inoculum in the next season. Causal organism. The causal organism of Molya disease of wheat is Heterodra aveni. Its systematic position is kingdom Animalia, phylum Nematoda, class Cicerentia, order Tylenchida, superfamily Tylenchoideae, family Heterodiridae, genus Heterodera, species Avinae. Heterodera avinae is an obligate parasite of cereal crops including barley, oats, wheat and rye. Cereal crops infected with this nematode are more susceptible to infection by fungal disease such as Rhizoctonia root rot. Morphology and life history of cereal cyst nematode. The cereal cyst nematode exhibits sexual dimorphism. The males and female nematode differ morphologically. The male is vermiform in appearance and transparent and measure 40 by 1300 micrometer in size. The female is rounded and white and usually vary between 0.55 and 0.75 millimeter in length. It is about 2/3 as wide as long. Sexes are differentiated at the third stage larvae. The males develop a single testis while the female have a paired ovaries. The fully developed female larvae of the fifth stage become typically lemon-shaped cyst. It develops egg that completely fills the body cavity. These adult females, also known as cysts, can be seen adhering to the roots and they gradually become brown mature cysts after the death of the female. In this picture, we will see the body of the mature female and the cysts which are present on the surface of the roots. The eggs are oval and the vermiform larvae molts 4 times. The cysts are typical lemon shaped. This is the picture of cyst. Cysts are actually the dead bodies of adult females which become dark brown in color. These cysts occur in soil and each cyst contains about 200 to 250 eggs. These are the eggs which are present inside the body of the female. And this dead body becomes the cyst. And this cyst contains the egg and it is a source of inoculum in the next season. These cysts can withstand harsh conditions and can be spread in soil or by agriculture machinery or by animals, wind or by dust storm or any other ways. Disease cycle. The disease cycle starts with the fallen cyst, which is the main source of primary inoculum. These nematodes survive in the soil by means of cyst. These cysts get separated from the roots and they may get fallen into the soil, and the eggs present inside the cyst do not hatch immediately. They require some time to mature. The intervening periods between the two crop seasons serve as a maturation period for the eggs and hatching of the second stage larvae. The first stage juvenile develop within the egg and after molting it gives rise to the second stage juvenile. The second stage juvenile is the infective stage and it comes out of the cyst and migrates through the soil in search of suitable host. After reaching the suitable host, this second stage larvae enters the host through the root tip. It usually enters behind the root tip and the juvenile enters through its by piercing through its stylet, and then it reaches the cortical region and or endodermal cells and near the pericycle cells where it feeds and the feeding by these second stage juvenile cause the enlargement of the cells. The group of enlarged cells are known as syncytia. The syncytia are the feeder cells. These are the feeder cells for the nematode. The second stage juvenile now then molts to become third stage juvenile, which on molting becomes the fourth stage juvenile. These fourth stage juvenile then develops either into males or females. The males come out of the root, and then these males then come out of the root and then enter the soil, move for some time, and then finally dies. While the female continuously lies inside the root and it either mates with the male or through parthenogenesis it produces the egg. The body of the adult female become lemon shaped, and these lemon shaped bodies are filled with the eggs. Uh, by the end of the plant growth season, the body of the adult female turn into brown cyst, and its external surface hardens and her internal parts dies. These cysts then attach to the root surface of the host plant, or they may fall into the soil and serve as a source of primary inoculum for the next growing season. In this way, the disease cycle completed. Control measure. The disease can be controlled by several means. First, plowing. Plowing the fields 2 or 3 times in the month of May to June definitely reduces the primary inoculum, because the cysts are very susceptible to desiccation and cannot tolerate hot sun and hot summer winds. Second method is crop rotation. It is recommended that wheat crop should be grown in rotation with non-host crop such as gram, carrot, radish and resistant varieties of barley for 1 and 2 years. Since the nematode is highly host specific, long rotation of crop at least of 4 years are more effective for controlling the disease. If a non-host crop such as chickpea or mustard is sown for 2 to 3 consecutive years before the host crops are planted, a profitable host crop can be raised in the field. Fallowing of the field. Fallowing of the field for 1 year can reduce the inoculum by 47 to 55% and increase the crop production by 85%. The fourth control measure is green manuring. Green manuring of infested soil with chopped cabbage leaves heavily reduces the larvae in the soil, and this practices increases the activity of nematode trapping fungi such as Arthrobotrys oligospora and Dactylaria thomassia in the soil. Chemical control is also an effective measure to control the disease. The soil application of LD carb at the rate of 2 kg per hectare result in significant control of the disease. Another chemical control is the fumigation of soil with DD mixture normally kills about 90% of these larvae present in the soil. And this treatment is effective for short time because the remaining larval population multiplies rapidly in presence of the susceptible host. Use of carbofuran at the rate 1.5 kg per hectare in the soil also control the disease to the greater extent. Use of disease resistant variety. Raj MR1 is the resistant variety of wheat which can be grown in Molya infested fields. BH75 and BH393 of barley can be grown in that area where wheat cultivation is not possible due to heavy damage. Early sowing of crop. The crops may be planted up to 15th of November. At that time, the soil temperature is low and the nematodes are not active. The early plantation of wheat crop can escape the nematode damage since the crop is sufficiently healthy to tolerate the nematode attack. For further reading, I would like to suggest the following links and a book of plant pathology by George and Agrios.